So hello everyone. Today we're going to talk about vacuotiners. See, vacuotiners is nothing but they are the colored plastic tubes. They use it to collect blood samples and they containing different types of additives or clotting factors in it. And we are going to the laboratory for further evolution like blood investigations. So these are the different colors of blood containers, the most commonly used in the medical field. First, we will understand the general concept. When we take a blood, there are two components are present. One is cellular component, another one is the plasma. Cellular components contains RBC, WBC like platelets, and the plasma components contains water, electrolytes, hormones, enzymes, protein factors, glucose, proteins, urea. So these are all the components present in the blood. So for analyzing the each components, analyzing each component. We are using different colored tubes and they are using different coagulative factors in it. Okay. Then another one thing, serum. You know what is serum? Usually, blood contains. We have seen blood contains cellular component in the plasma. So from the plasma, if we remove that clotting factors, it is called serum. So this is only serum is plasma minus clotting factors. Already told, we have seen the uses. So the vacuotiners are used to collect the samples, like blood samples from the patient, and they used to transport and the processing of blood samples for diagnostic purpose in the laboratory to identify cellular component levels like hemoglobins, RBC, packed cell volume, complete blood count. So these values are going to identify in the laboratory. Okay. First, we will now see the types of vacuotiners. So first see the red vacuotiner. Red vacuotiner actually it contains no additives. So it doesn't it doesn't contain any clotting factors in it. So it is used for analyzing like fasting blood sugar, RFT like renal function test, like LFT liver function test, and serum electrolytes and viral markers HBsAg, HCV, and VDRL. So these are the few components they use they are identified by using red containers. Okay. Now see the purple container. Okay, purple. The additive used in purple container is potassium salt of EDTA. We know what is EDTA. It is ethylene diamine tetraacetic acid. Okay, they use it for analyzing whole blood like CBC, blood glucose and typing. I'm sorry, blood grouping and typing, and also used for blood smear like peripheral smear for malaria. Okay. Now see the green vacuotinner. The green vacuotinner, the additive used is sodium heparin or lithium heparin. So these anticoagulant the binds with the clotting factors and they inhibit thrombin and thromboplastin, so that only there will the no clotting has occurred. So this heparinized whole blood they are used for collecting plasma samples and for electrolyte analysis. Okay. Now see the yellow vacuotiners. The additive used in yellow vacuotiner is trisodium citrate with acid citrate and dextrose. So these are the anticoagulants used in the yellow vacuotiner, and they are used for tissue typing, DNA study, and also used for HIV culture. Okay. Now see the blue vacuotiner. Sorry. The additive used in blue vacuotiner is sodium heparin or EDTA. Actually, this is the after after taking blood, first we will draw the blood in that culture tube, and then next to the culture tube we are going to fill the blood in the blue container. It contains sodium heparin EDTA. They used for coagulation studies like Bothra time and activated partial thromboplastin. Okay, this is used. And gray container. The gray container, the anticoagulant used is potassium oxalate and sodium fluoride. This potassium oxalate binds with the calcium and they inhibit coagulation, and sodium fluoride. This sodium fluoride inhibits glycolysis. See, this is glycolysis pathway. Then this is sodium fluoride. This sodium fluoride this inhibit the enolase enzyme. So the glucose is not metabolized. So glycolysis is inhibited. So we will find the blood glucose level, like plasma glucose, blood alcohol, and lactic acid level. So these are the uses for gray container. First, in what order we are going to draw the blood? Let's see, order of draw. So after taking blood from the patient, first we have to fill the blood in the culture tube, because so that only we will minimize the of contamination, so that only the result we can get may almost positive, like reliable result. And then we have to fill the blood in the sodium citrate, like blue container, okay, for coagulation profile. And then we are going to fill the blood in the plain tube, like red tube. And then we are going to fill the blood in the green tube containing heparin, okay, then which is used for electrolyte analysis, plasma samples analysis, okay. And then we are going to fill in the purple container, lavender EDTA, which contains EDTA used for complete blood count. And then finally we are going to draw the blood in the gray container, okay. It is used for analyzing plasma glucose level. Thank you.